So now that we've talked about some of the ways you can clone a DNA segment or a gene, now we can talk about some examples of things that you might want to do with your cloned DNA or with your recombinant DNA. And there may be situations where once you clone the DNA, just making copies of it or studying the DNA sequence directly may be all you're trying to do, but there may be situations or experiments where you might want to alter the gene or the DNA that you've cloned in some way and see how that affects its function. So again, this is something you might do with a gene that you've isolated from, say, human DNA or some other eukaryote, but you want to study how it behaves. So you clone it into maybe a bacterial vector, or it could be a yeast vector or any other host organism. So then by altering that gene, and especially for an expression vector, you can see how a change in the DNA sequence will affect maybe the function of the or the structure of the protein or the way it's expressed or whatever you're trying to determine. So if you want to alter the gene you've cloned, there are a couple ways to do that. One, if you have an altered version of the gene, maybe from a mutated form of the gene that you've isolated from a different wild type strain, or a sequence that you've artificially mutated by changing the base sequence, or maybe even a synthetic DNA segment that you've created with a mutation contained in it. If your clone DNA is inserted with a restriction endonuclease, you can just cut out the original version and replace it with the mutated version. You can even do this as long as the mutation site is flanked by restriction sites that are conveniently placed. You can add these anywhere you want in your clone. On the other hand, if you don't have a clone with a convenient set of restriction sites flanking it, you can introduce a mutation and along with, including a restriction site, using what's called oligonucleotide-directed mutagenesis. So remember, oligonucleotides are just short sequences of nucleotides, maybe 10, 15 base pairs. And so that's the same size roughly as the primers used for PCR. So if you have a plasmid containing your cloned DNA, but you want to target a mutation to a particular region within that clone, then you can use primers that are have the same sequence as your gene or the target region that you want to mutate, except for maybe one or just a few bases that introduce a mutation. Now, DNA, a single strand of DNA that's complementary to another strand will hybridize, meaning that they will hydrogen bond with each other. But even if if you have a sequence of 10 or 15 bases and only one or maybe two of them are different, they will still hybridize, just not quite as well. So you can introduce primers that contain a mutation. So this little X would be a spot where the DNA sequence is slightly different, maybe just one base different from the original sequence, and it will still hybridize. So you can do PCR with these sequences the same way you would with regular primers. So you denature the DNA of the plasmid, you let the primers anneal, and then PCR, you can do PCR to copy the entire plasmid because they're circular. Of course, the DNA polymerase will just copy the whole thing until it gets back to the other end of the primer. Again, separate out the now copied plasmids, let them anneal to each other because now they're completely complementary to each other, but now they contain your mutations. Now you have a complete plasmid that contains your DNA of interest, but now with some sort of DNA change. So again, it could be just a single base or something more complex depending on what you're after. Then you transform that into a host cell. The host cell usually will repair the nick that's left over, so there's still these gaps that are present because the PCR enzyme will not repair those gaps, but the host cell will have enzymes that can do that. And these are just examples of how you might introduce a mutation into a cloned gene, and then you can study the effect of that mutation in your host cell. So maybe if it expresses the protein, you can isolate the protein and see how the amino acid sequence, changing the amino acid sequence has changed the structure, or you can look at how then the protein binds to other proteins or whatever you're trying to get to. Now the other nice thing about cloning is that you can clone together DNA segments from any number of different sources. So you don't have to just clone together a gene and a plasmid. You can take a gene and a plasmid and another gene and fuse them together. So when we talk about gene fusion or fusion proteins, those are formed from recombinant plasmids that contain the DNA for two different genes that are attached to each other in the plasmid end to end. So there are lots of ways to do this, and lots of reasons to do this. One example is a type of purification that you might do called affinity purification. Now, we've talked about affinity purification before. So if you have a protein that binds to a specific ligand, then you can put the ligand on, say, a column, and you can run your protein mixture through that column, and your protein of interest will bind to that ligand. But there are lots of proteins that maybe don't have a specific ligand, or maybe the specific ligand is too difficult to immobilize on a column, or there may just be some reason why you can't directly purify that protein using affinity purification. So instead, what you can do is make a fusion protein between your protein of interest, your target gene, and another protein which has a ligand that is easy to isolate, easy to immobilize, and therefore easy to do affinity purification. And so we take those two proteins and make a fusion protein. So an example of that is this enzyme called glutathione S transferase. Now, the details of this enzyme aren't important, except that its substrate is this substance called glutathione. So glutathione is a short 3 amino acid molecule, and it binds directly to this enzyme. So to make a fusion protein, you make a recombinant plasmid that contains the DNA for the gene for the protein that you're trying to purify, and the gene for whatever protein you're going to use as your purification tag. So again, glutathione S transferase is a common one because it's got an easy ligand to use in purification columns. But you could use other ones. So you take the DNA for both. They're both cloned into the same plasmid, but importantly, they are cloned back to back. In other words, or end to end, meaning that when one of them is transcribed, they're both transcribed into one mRNA, and then the mRNA gets translated into a single protein that has the amino acids for both, in this case, GST and your target protein. So then you end up with this fusion protein, which contains, sometimes these are called chimeric proteins, and so they have a fusion tag, but also the protein you're trying to purify. So you then grow these cells, your host cells, bacteria, whatever they are, and extract the protein from the cells like you would any other protein. But then you run that extract over a column that contains beads, which have your affinity tag protein ligand bound. So in this case, if you're using GST as your tag, then your beads would contain glutathione, or would have glutathione attached. So the idea is that the other proteins and other things that aren't actually don't have GST bound to them will run right through the column, and then your protein of interest and the glutathione enzyme they're attached to will stay on. Now the nice thing about this is, again, you can use affinity purification with the protein itself if it happens to bind to a ligand, but maybe your protein of interest doesn't have a good ligand that you can use, or maybe it's too non-specific. So maybe there's a bunch of other proteins that might bind to your protein. In this case, you're using glutathione, which is very specific, and importantly, it's not an enzyme that would be found normally in your host cell. So that means that there would not be any likely to be any protein normally expressed by your host cell that would bind to the ligand. So it's a very nice way to make sure that only the protein you want flows through. And then, of course, to get the GST bound protein off, the same way you would with a regular affinity column, then you run the free glutathione or whatever your ligand is over the column. That will bind to the GST and then remove it from the beads, and so it will then elute through the column separate from all those other proteins that you don't want. Then once you have your fusion protein, you can do whatever you want with it. You might be able to study it as it is, or you can remove the GST tag, usually with some sort of peptidase, some peptide cleaving enzyme. Now, another kind of special fusion protein you can make are reporter proteins or reporter genes. So proteins, of course, most of them are not visible, meaning that by themselves they're too small to see with the naked eye, and even under the microscope, even at high concentrations, they're very hard to see. So detecting proteins, specific proteins especially, is difficult. But there are some proteins that have the ability to be visible under the right circumstances. So there are, for example, a number of different fluorescent proteins. So a fluorescent protein or a fluorescent molecule is something that when it absorbs light at a certain wavelength, it emits light at a different wavelength. And so basically that usually means, and the release of the light can be kind of delayed. So those proteins are often able to kind of glow in the dark. So a lot of organisms actually express these kind of fluorescent proteins. A lot of invertebrates that live deep in the ocean where there's very little light will actually express these proteins. Usually they use them to either communicate with each other, or they may use them to startle or confuse predators. But in any case, we can get the DNA for those genes. So for example, the first one discovered, and the most famous one, is called green fluorescent protein, or usually it's just called GFP. And this is what GFP looks like. So it's a protein, and it has this fluorophore molecule. So there's a aromatic molecule that it carries around that is fluorescent. So again, this protein is normally expressed in certain cells in jellyfish, but we know the DNA for it, so we can make fusion proteins that have a GFP tag, which means that they express GFP in tandem with whatever protein we are interested in. And there's, like I said, a bunch of these now. So down here you can kind of see this is the whole rainbow of colors that you can use for fluorescent tagging. And so with the fusion protein, then you can express a chimeric protein that consists of your protein of interest with a fluorescent protein attached to it. And then you can look in a microscope and with a fluorescent light, or with the right filter, you can see the light fluorescing from these proteins. And that will tell you, especially if you're looking at a cell in culture or a piece of tissue from an organism that's been genetically modified. So over here, these are actually bacteria that have some sort of fluorescent protein expressed. It looks like something that's actually attached to their DNA, to their plasmid. But you can also use as a host for your recombinant plasmids or recombinant DNA any organism, hypothetically. So in fact, a genetically modified organism is just any organism in which the animal has DNA that is either a modified form of its own DNA or DNA from another species. And so this over here, for example, is a nematode worm, commonly used model animal in a lot of developmental and genetic research called C. elegans, and it has been modified to express at least one fluorescent protein, looks like maybe two, in a subset of its cells. So what they've done is the protein being expressed only shows up in certain cells because that protein, the protein of interest, is only normally expressed in those cells. So it's a way to just visualize where a protein is expressed in a cell or in a tissue. There's another form of reporter gene that is not inherently fluorescent. So GFP and others like it are inherently fluorescent because they have this sort of modified, at least in the case of GFP, it has this modified amino acid or form of serine that is fluorescent. So this covalent modification happens in this protein after it's synthesized, and so this fluorophore that it carries around is just part of the protein. But there are other fluorescent proteins that do not have fluorophores themselves, but instead act as enzymes that can produce fluorophores or fluorescent molecules. So an example of one of those is called luciferase. So luciferase and other enzymes like it are not fluorescent by themselves, but become fluorescent, or the fluorophore they produce becomes fluorescent. So you can turn them on and off by having the substrate present. So you can design all kinds of nifty little fluorescent constructs using different combinations of fluorescent proteins or fluorescent enzymes to ask different kinds of questions about the location or activity of your protein. And again, this is just another example. This is another worm up here where, again, here they don't even have, it's not really even a fusion protein. So here it's just the GFP, and then what's called a cDNA. We'll talk about what that means in a minute, but basically it's the mRNA for, again, some other gene, but then it uses the same promoter for that gene, so it only shows up in the cells where that protein normally would be found. But you can even construct these without the cDNA, without the DNA for the protein, so you could just have the GFP and the promoter, and it would just show up in those cells without any fusion protein at all. Now, another use for fusion proteins is to look at protein-protein interactions. So if you have, there's lots of questions you may ask about a protein once you've isolated it, whether you've isolated it through cloning or not. But one thing that you usually want to know about a protein, if you're wondering how it works, is what other things it binds to, especially other proteins. So you can use fusion proteins with the to create a construct that you can then look for other molecules that bind to it. So this is actually kind of like affinity purification, but and this process here is called immunoprecipitation. So your vector will contain, again, a promoter, which is a DNA sequence that will tell whatever comes after it to be transcribed and then translated. So your cDNA will be then the DNA for the protein that you're interested in, and then more DNA for a protein which is going to be an epitope for an antibody. So we talked about antibodies before. Antibodies bind to usually molecules that are from infections like viruses and bacteria, but you can make antibodies against anything artificially, and the thing that the antibody binds to is the epitope. So if you have an antibody that has a known epitope, then you can use the epitope as kind of an affinity purification tag for a promoter. But here what we're doing is we're growing now this fusion protein. So this is the fusion protein, which contains the amino acids for our protein of interest, plus this epitope tag, whatever it is. Here, it's not a specific enzyme substrate interaction or receptor substrate interaction. It's just between the antibody and the epitope. So you grow this fusion protein in cells, you extract protein from the cells, and then you mix the cell extract with the antibody for that epitope. And what will happen then is the antibody will bind to that epitope, form these large complexes that then precipitate or can precipitate out of solution. So we call that an immunoprecipitate in this case, because it's being precipitated by the presence of these antibodies. Now, the precipitate is easy to isolate because now it's a solid, so you can use centrifugation, for example, to isolate it from anything that doesn't precipitate out. And then you can separate the antibody from the epitope and from other proteins using a gel. And the reason for doing this is that when you precipitate out the protein of interest with your antibody, you will also precipitate out any other proteins that might have bound to your target protein. So, in your cell, there could also be other proteins attached to the target protein. And so when you run the immunoprecipitate out, you will get other bands that come with it. And the assumption is that those bands represent new proteins, or proteins that you just discovered have the ability to bind or interact with your protein of interest. So this is your protein of interest, and that's what should have come out, because again, that's your fusion protein. But then these other bands represent proteins that interact with your protein of interest. So now you've just identified a new protein interaction. This is another type of fusion protein that you can use to study protein interactions and to purify. So here, kind of like the affinity purification step, our fusion protein consists of our target protein. This is the thing we're studying, and it's bound to this tandem sequence of proteins. In this case, they've used this one called calmodulin. And then this linker region, which is, as we'll see later, has a site to be cut by an enzyme called TEV protease, and then another protein called protein A. So again, the idea is you mix together this protein, this fusion protein, with a crude cell extract. So this is just a mixture of proteins from a cell or a tissue extract, and what you're looking for is proteins in this crude extract that might bind to our protein of interest. So that's what we want to know. Are there any proteins in our cell or in our tissue that binds to our protein of interest? So when we mix them together, if there are any proteins that bind to our protein of interest, they will stick to it. Then you run the whole thing over a column of beads. In this case, we're using the IgG, those are antibodies which will bind to, in this case, protein A. And then after we run that out, all the things that don't bind to our protein of interest will just run through. And so on our column, we will still have our fusion protein plus anything that does have a high affinity for, or a reasonable affinity for our protein of interest. But then to get them off, to get the fusion protein off of these beads, then we use this enzyme called TEV protease. And a protease is just a protein that cuts up other proteins, and this particular one happens to have a cleavage site within this little linker region, so it just cuts it right there. And then that releases it from the column. So now on the column we have our protein of interest attached now to just the calmodulin binding protein. And then our protein interactions here, the other smaller proteins that bind to our protein of interest. So we run that over a different column. Now instead of IgG beads, we have beads containing the protein calmodulin. And so now our protein of interest will bind to these calmodulin beads because it is fused to a calmodulin binding peptide. And now we then have our protein on there with our target protein of interest with whatever is bound to it. We can run the elute more buffer over the column, and the first things that should come off are any proteins that are loosely bound to the protein. So not all proteins, or not all protein interactions are the same. Some are stronger than others. So weak interactions or proteins that weakly interact with our target protein will come off first, and then finally we elute the target protein itself, and whatever's left over will be just those proteins that specifically and strongly bind to our protein of interest. And then here we have yet another fusion protein that we can use to look at protein-protein interactions. This one's a little more complicated. It's called a yeast-2 hybrid analysis. But again, the question is the same. You have a protein, and you want to know what other proteins interact with it. So in this case, we're going to make a fusion protein that has our protein of interest, protein X, and then fuse to that a regulatory protein. So a protein that's going to bind to the DNA of a reporter gene in yeast. So it's this gene called Gal4P, and the Gal4P DNA binding protein or DNA binding domain will bind to the Gal4P DNA element in the yeast chromosome. And then we have we clone into this same cell a different fusion protein, one for another protein. So our question is, do protein X and protein Y actually bind to each other? And so protein X will have the DNA binding domain of this Gal4P protein, but then protein Y will have the activation domain for Gal4P. And so this Gal4P is a protein that normally would come together and act as a transcription factor for certain genes in the yeast cell. So these yeast will have some reporter gene which is under the control of this Gal4P promoter. So this gene will only be expressed if these two proteins, the Gal4P activation domain and the Gal4P binding domain, are close enough together to recruit RNA polymerase. And we'll talk all about RNA polymerase later on, but that's the enzyme that's going to make RNA. This is the mRNA coming out here for whatever our reporter gene is. So the reporter gene could be maybe a fluorescent protein. Maybe in this case, what they've done or what they could do is use an enzyme that turns blue. It doesn't matter. Whatever the reporter gene is, it just tells you that in this cell, these two proteins, X and Y, got close enough together that they were able to activate this gene. If protein X and protein Y didn't bind, then you wouldn't see a reporter gene being expressed. So again, you combine your two strains, each of which has its own recombinant plasmid, its own fusion protein. Yeast do reproduce sexually, so you can combine two strains together and they will have offspring. And so you look again in the offspring to see which ones have the reporter gene. Now here they've used some sort of negative selector or positive selector protein or survival gene, which so that means that only in those cells in which there was interaction will the colonies actually survive. And then you go back and look at the fusion proteins to figure out which ones are actually interacting. Now, of course, there are plenty of other examples of gene modifications that we could talk about that are interesting, but a particularly interesting example of genetic modification that is becoming more and more important these days involves this system called the CRISPR-Cas9 system. And basically, this is a system that evolved in bacteria probably as a sort of immune system. So in your immune system that if you get infected with something, your body, your immune system has the ability to remember what you were infected with later and respond if you get infected with that same virus or bacteria again. Bacteria have to deal with infections too. They get bacteriophages, and those can kill them. So bacteria have evolved a number of defense mechanisms, and the CRISPR-Cas9 system is an example of that. Basically, the CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats, which is just a name for the type of DNA segments that this type of the DNA elements that the bacteria express that they use to recognize these sites. And they're basically DNA in the bacteria that are actually derived from viruses. So the bacteriophages sometimes, when they infect, if the bacteria survives, some of the viral DNA can actually end up in the bacterial genome. And so they end up as part of these CRISPR sequences. And then Cas9 is the enzyme that recognizes these sequences and can cut them in the viral DNA. And so the way Cas9 works, it's a complex actually of several different proteins, but it carries around with it a molecule of RNA called the guide RNA, or here it's called the sgRNA. And that RNA normally in the bacteria is encoded by the CRISPR sequence. So the CRISPR DNA sequence is transcribed into the guide RNA. The Cas9 enzyme carries the guide RNA with it, and it looks for DNA that it's complementary to, which again should be in the bacterial cell would be the viral DNA that it got infected with. And then the CRISPR-Cas9 recognizes that DNA because again it's going to match the guide RNA, and it will cut the viral DNA, therefore hopefully preventing the virus from replicating. So what we can do though is we can take that property and we can make basically, because the Cas9 enzyme will carry around any RNA sequence as long as it has the kind of recognition. There's part of the guide RNA that the Cas9 enzyme recognizes, but the part that it actually uses to detect its target sequence can be any arbitrary sequence we want. So to use CRISPR to modify DNA, you make a CRISPR-Cas9 plasmid, which contains the DNA for all the Cas9 proteins, plus the DNA for our guide RNA. And then we put both into a host cell. So again, the plasmid has all the things we need to make the Cas9 protein and a guide RNA. And you put that into a cell with your recombination fragment, which is your target DNA, the thing you're trying to interact with. And then the host cell will make the Cas9 protein and will transcribe the guide RNA, and then the Cas9 protein will carry the guide RNA and start essentially reading along the DNA of the host until it gets to the target sequence. And then when it does get to the target sequence, it will cut it. So as it moves along, when the target sequence, which is part of the guide RNA, which again is we artificially put in there to specifically look for our chosen sequence. When it gets there, the guide RNA will bind to the target DNA, and then the enzyme will cut the DNA. Either depending on what kind of Cas9 you use, it'll either make a double-stranded break, or there's a version where one of the nuclease domains it becomes inactivated, in which case only one strand gets cut. If you're cutting both strands, then you create what's called a non-homologous break, which means that both strands are cut. And so most cells have mechanisms for repairing non-homologous cuts in double-stranded cuts like this, but usually they're very error prone. So after the cut gets made, the repair mechanism will try to put the ends back together, but usually it replaces, again, because there's no way for the enzymes to know necessarily what bases may have been lost. Some random bases get added or get changed, and so you're using essentially the CRISPR-Cas9 system to introduce a gene mutation into a particular sequence. So usually this is done to just turn off a gene in a cell, but it's a very easy way to selectively target one particular gene. But you can also use CRISPR-Cas9 to add genetic sequences into a genome. So in that case, you combine your CRISPR-Cas9 system, again, with one of the nuclease domains inactive, which means it just doesn't cut. So only one of the strands gets cut, but this time you've combined your CRISPR-Cas9 plasmid with a restriction or recombination fragment, so a DNA sequence that you want to add into the cell. And that DNA sequence will be homologous or partly homologous, with one of the strands of that you cut. And that will again trigger the DNA repair mechanism in the cell, but this time, because there is only one cut and because there's another homologous strand, some of the copies of that gene will be repaired by replacing the cut DNA with the fragment that you inserted. So you can essentially replace the target sequence with your DNA of interest. So this is how you would use CRISPR-Cas9 to introduce new DNA into a segment. So again, this is an easy, relatively easy way to either mutate genes or add in new genes into a genome by cutting the DNA. And then you can also do some even more complicated stuff with this. So a really interesting way to use CRISPR-Cas9 is this gene drive system. So the gene drive system includes not only the CRISPR-Cas9 DNA, but also the DNA. So the whole plasmid contains, again, the Cas9 enzyme, the guide RNA, which is going to tell the Cas9 where to cut, and then your so-called payload gene, which is the DNA you're trying to introduce into the cell. So all three together, rather than have the recombinant DNA separate, can then be inserted into the genome. So now you add this as a plasmid, but then usually there's two homologous regions. And again, homologous DNA means it has the same sequence, essentially, in this case. So the DNA gets cut. Again, you're creating a mutation, but because you also have those two homologous regions, the cell's DNA repair mechanisms will recognize the plasmid itself and will incorporate the plasmid DNA into its chromosome. So now you have a chromosome which actually carries with it the Cas9 DNA, the guide RNA, and the payload gene, which means that not only will the payload gene be present, but the machinery necessary for making more Cas9 cuts will be present. And the reason that that's important, and you can either do this with a cell that and you can have it such that both copies of a gene get replaced with the payload gene in this case. So again, if you're an organism that reproduces sexually, you usually have two copies of every gene, and so by doing it this way, not only do you change one copy, but now the cell carries around everything you need for the cell to make its own modification to the other copy. And then when that individual reproduces, that means that it passes this now modified, this new Cas9 CRISPR complex onto its offspring. The offspring will not only inherit that gene, but will actually then copy it and make sure that both copies are passed on. Now, we haven't really talked much about genetics in this class, but you probably know, again, that everyone carries two copies of every chromosome, and when you have offspring, only one of those chromosomes gets passed on to your offspring. So if you are heterozygous, that means you have two different versions of the same gene. One you got from your mom, one you got from your dad. When you have kids, only one of those usually gets passed down. So that means for any given individual, only about 50% of its offspring will carry a given copy of a gene that it has. And then its grandchildren will only have a 25% chance of inheriting that gene, and so on. So that means with every generation, it becomes less and less likely that any given individual will inherit that mutation. So if you want to introduce a genetic modification into a lineage, in other words, not just an individual, but also its offspring, then you have to basically do a lot of breeding in order to get a homozygous individual, one that has two copies of the gene, and then mate it with another homozygous individual just to guarantee that all of the offspring have that trait. But with the gene drive system, if you have one individual that carries around the gene drive complex, then it will pass, 50% of the offspring will inherit the gene, but then because the gene drive carries everything it needs to make a copy of itself on the other chromosome, if this individual is homozygous for our modified our mutation, and it's mated with an individual that's homozygous for the wild type version of that gene. Again, normally that would, over here in our normal inheritance pattern, if we did that. So our heterozygous and our homozygous individuals. Actually, let's do it the other way around. Let's make it so that just to make it simpler, we'll say that the red individual carries a dominant allele, which means that only needs one copy, and let's say that it's heterozygous. So that's what that big A little a means. It has one dominant allele, one recessive allele, and the other parent is homozygous for the recessive allele. So that means that half of its offspring will also be heterozygous, and then half will be homozygous. And so that in the offspring that was homozygous, essentially the pathway has been cut off. And so again, each successive generation will have less and less of a chance of inheriting it. Now over here, though, with our gene drive system, again, even if the parent can't be heterozygous, it has to be homozygous because its gene drive system will make a copy of itself on the other chromosome, and then that means that both of its offspring will inherit that allele, and it will also inherit the recessive allele from the other parent, but the gene drive system will modify the other chromosome so that it becomes homozygous. And that will be true of the next generation as well. So the grandchildren of that first generation will also then become homozygous for the mutation with the gene drive component. So what that means is after a few generations, because 100% of the offspring from anyone who carries that mutation will carry the new mutation, eventually in a population, wild population or a lab population, the entire population will carry that mutation. This technology has been kind of explored for example, treating things like malaria. So malaria, you probably know, is carried by mosquitoes, but the mosquitoes themselves, if you can genetically modify them so that they can't carry the malaria parasite, you could modify one individual normally, and then hope that individual has a lot of offspring and hope that gene spreads just through normal inheritance. But if you instead use the gene drive system, you can guarantee that after a few generations, all of the individuals in all the mosquitoes in a population will carry the gene that prevents them from spreading malaria. So there's lots of kind of interesting techniques or applications for this kind of technology. All right, we'll stop there, and then the next video we'll talk about what you can do with genomic information.